Bem, dando continuidade então ao
Well, moving forward with our program, we invite as chair of the panel the Vice Director of Bio-Manguinhos for Innovation, Ricardo de Godoi. For the presentation, online presentation, we invite the senior scientist for the bioengineering for Max-Planck-Institute for complex systems, Yvonne Genzel, who will give the presentation Next Generation Virus Production from Clone to AMBR to Perfusion Very High Virus Yield. She is a doctor in biocatalysis by the Mediterranean University in Marseille, France, and also postdoctorate in Von Goethe University in Germany, where she acts as a professor. And in the Max Institute, she leads the upstream processing team with the production with mammal cells. The Vice President Associate of Strategy for Investment and Development of Portfolio and Innovation of Global Health, Innovate Technology, Hayato Urabe, who will make the presentation Vaccines for a Safer World, the Pitfalls in the Development of Vaccines for Neglected Diseases. He concluded his bachelor in molecular biology, has a master of entrepreneurship engineering, has a doctorate in biomedical engineer from Brown University. He has an additional master's degree with global policies in University of California, San Diego, and he acted in finance and technology areas and strategic planning and due diligence of startups and project management in the Silicon Valley. He was a consultant for synthetic genomics for technology that goes from nutrigenomic until biofuels. He oversees all the activities of investments and access, such as exploratory activities to seek for technological seeds to apply to global health and its financial models that are innovators. We invite to the stage the professor in technology innovation projects from Getúlio Vargas Foundation, Paulo Negreiros Figueiredo. He will make the presentation, Managing Innovation in Human Health Biotechnology Startups in Brazil, Evidence and Lesson Learned. He's a doctor in Science, Technology, and Innovation by Success University in the UK. He's a senior researcher at Oxford University and a founder and responsible for the research program in technology and innovation of Getúlio Vargas Foundation. So I pass the floor to Ricardo Godoi. So good morning. I'm here again with you. I was here in the previous session, and I would like to thank the presence of the guests here. I had the opportunity to talk a little bit with Yvonne and Hayato online, and I would like to thank we have a speaker from SGV in this space that is being very good for a symposium. This is a subject that has gained the visibility and attention and regarding innovation for to the government for the efficacy that is expected, and we have given a very fair amount of attention for this subject, and also we have been working with implemental science. And I would like to confirm with the speakers if the translation is okay, if you are receiving the translation. Are you able to understand? Because we speak English, but I know you have interpretation, so I can see that you can understand me. The translation is okay, right? Can you confirm, please? I think that Yvonne may be...
Tudo bom.
Tudo bom. So thank you for joining. Good evening for you. I think it's good afternoon for Yvonne. And it's a pleasure.
We'll just pass the word to Yvonne to start. Yvonne, please, if you can jump in. And I will take a close look on the time so we can have more time to discussion with the audience here. Thanks.
So thank you. I hope you can see my screen. I have no view on what it does right now. Can you see the screen, Ricardo?
Yeah, we can see the screen.
Okay, thank you. So I would like to start by thanking the organizers for giving me the opportunity here today to give you my idea of how I see that we can overcome the valley of death. I will start, by explaining that, well, how this goes for virus and vaccine production. So at the beginning, you have a virologist or a medical person who has an idea for a new vaccine. He's producing this in a small scale, typically with adherent cells in a tea flask, and he's happy to have 1 × 10⁶ infectious variants per milliliter. Then he tries first experiments in mice. For that, he needs to purify the material, and this is typically easily done in centrifugation. And then comes the next step, and this is what everybody always refers to, the valley of death. You have the idea, and now make it to a product. So what you will need an easy, scalable, GMP compliant process, and you are targeting to much higher virus concentrations. So for getting into vaccine doses of 1 × 10⁷ or 10⁸ variants per dose, or for gene therapy and virotherapy even higher, you are aiming for producing in your bioreactor 1 × 10⁻⁻⁰ infectious doses typically. For that, you will need a high producer cell line, and doing this with your adherent cell line that you have in your lab will probably not be okay, because you need a qualified GMP-ready cell line. And if possible, it should be a suspension cell line. So the technologies today are emerging, and we have already heard it in the previous presentations this morning on what great laboratories that are there at the CNPM to be there. So I would like to give you a flavor that actually I see it not as a valley of death, but a sea of opportunities that we have today with all the technologies that are available. So if you think of producing a vaccine with adherent cells, you grow the cells typically to 2 million cells per ml, and then you infect them with virus. If you do this with what we heard this morning with adherent viro cells and on microcarriers, there you do typically also medium exchange. And yeah, if you do this with suspension cells, your starting material comes from shake flask, and here this is already good. You can today grow these cells up to 10 million cells per ml. With the new packed bed bioreactors available, you can, with adherent cells, easily also reach higher cell concentrations than that with microcarriers. Disposable bioreactors available today are 4,000 liter ones, and for example, also orbital shaken bioreactors up to 2,500 liter scale. But also microbioreactors are there, and this can ease up your process development. So these are small bioreactors of the size of 15 milliliter working volumes, and you can run 48 bioreactors in parallel. On top of that, there is now also automated equipment to do clonal selection. This is not so much done in virus production yet, but for monoclonal antibodies, cell line development, this is used a lot. And I would like to demonstrate to you on what this can do. And finally, what I want to show you here as well is that you can today also grow suspension cells with the respective cell retention devices easily up to more than 100 million cells per ml. Why is this important? If we look at the viruses that can be produced, So the ones that we work in our lab mainly, for example, influenza, modified vaccinia ankara, yellow fever or Zika, also a vesicular stomatitis virus. You see they all have different shapes, different sizes, they have a different genome, they replicate differently. But moreover, if you look at the cell-specific virus yield, so how much the virus produces per cell, you can already see that there's also quite a difference. So in respect of infectious particles, for example, yellow fever, if you are happy, you get 20 virions out of one cell line, of one cell. So, and at the end, you need to match the cell line you're using with the virus that you want to produce, and not every cell is producing every virus. So we are lucky with a long collaboration record with many, many partners. We have been now collecting several suspension cell lines in our group that you see here listed. And if you now take such a microbioreactor and you do parallel experiments, you can do a quick screening of all the cell lines you have available. And this is what you see here. So we have tested for a yellow fever construct all the cell lines we had, and the reference in adherent vero cells is the red line here. You see that already by screening without optimized condition, we were doing better than the reference. We continued by taking one high producer, one middle producer, and one low producer cell line, and tried to add viral sensitizers. These are small compounds that you can add at time of infection, and they have an impact on the virus yield. But you need to screen them. So we did a screening of 8 different compounds together with the company Verica. And here you can see that we were able to increase the yield even more. So first of all, 100-fold increase by just screening, and then another more than 3-fold higher. I said I want to show you about the clonal selection. So this was a cooperation with Sartorius where they did the clone generation, and they gave us 10 different clones to compare. You see here what the machine is doing. So you really start from one clone, and then you expand them, and then you can test it. And we had the task to test these cells for influenza virus production. We weren't sure if this was really possible to be able to see which one was best. So we first performed shake flask experiments just to see on the growth. So some of the clones were just reaching maximal cell concentrations below 5 million cells per ml, but some were really outperforming, reaching up to 15 million cells per ml. So really high growth. When we then tested in the AMBR microbioreactor again, for virus production, here you see the infectious virus titer and the HA virus titer, which is the important one for influenza. For total virus yield, you could see that the clones were doing quite different. And if you try to put this into numbers and compare this, so here we look again at the cell-specific virus yield here in total virus particles, you see that one clone was really outperforming all the other clones. So it makes sense to do something like that to already give you a good starting material. Now trying to grow into high cell concentrations. Today there are several retention devices helping if you are growing cells in suspension to retain them in the bioreactor or to remove the medium and to give the cells fresh nutrients and fresh medium. Some of them are membrane-based and some of them are non-membrane-based. All you have the retention device attached to the bioreactor in the outside. Here you can see how this looks like connected to an orbital shaken bioreactor in our lab. This is a hollow fiber membrane that is doing the cell retention. And by using this membrane, we were able to grow avian cells from the company Probiogen up to more than 30 million cells per ml. In comparison, without the cell retention device, you see here the batch cultivation just of 1 million cells per ml. So if you now look at the cell-specific virus yield, it is important that you're able to produce the same amount of cells per viruses per cell as you can do without the cell retention device. And we could even show that we were outperforming and getting better cell-specific virus yields. And you can see that we were reaching very high yields, up to 1 × 10⁻11 total virus particles. We also tried this for another virus. This is for yellow fever and for Zika virus. This is also an avian cell line that we received from the company Valneva. And here we were also trying different perfusion strategies, so feeding strategies during this cell retention. And with our best run, we were able to grow these cells up to 160 million cells per ml. And this is the typical titer value that you get normally. And here you see that we were obtaining 1 × 10⁻⁻⁰ infectious virus particles per milliliter. And again, we were able to increase the cell-specific yield. So we think that with this system, out of 1 liter of harvest, we were able to produce 10 million yellow fever doses. Finally, I want to present to you a work that we have done recently on a vesicular stomatitis virus. This is a virus that fuses the cells before they lyse and before the virus gets out of the cells. So you get these big particles in your bioreactor, meaning that running this with a cell retention device, membrane-based, could give you some problems. This is in collaboration with a group in Munich, and they came to us and said, yes, we have the virus growing to 1 × 10⁶ in adherent cells. Can you try to bring it to 1 × 10⁻⁻⁰? We did a first screening, and in this case, the BHK21 cells were the ones that were performing best, and we optimized the whole process for this into a cell retention perfusion run, and finally used a new membrane, which is a tangential flow death filtration membrane, and were able to perform here also, grow the cells to 20 million cells per ml, and then finally almost produce 1 × 10⁻⁻⁰ virions per milliliter. So with over the years, with a lot of collaborations, we have been able to grow different cells that are good for virus production to very high cell concentrations. We have cultivated them in different bioreactor setups with different cell retention devices, and we tried different viruses. And as you can see from here, we are able to obtain very high virus titers by that. And so this is how we think that next generation virus production and vaccine production should go for. And I hope I have convinced you that all these new technologies really are giving you a new sea of opportunities. And what I heard this morning in the Innovation Hub, I think you are very well prepared in Brazil to take this route on and to make this happen. By that, I want to stop with my presentation and acknowledge all the cooperation partners and my team in Magdeburg. Thank you.
So thanks a lot, Yvonne. The session is structured in a way that we will go for like a technological improvement perspective. Then we will start to discuss a little bit more management and business strategies. So that's why we do have different topics. And it was great to have your presentation, Yvonne, that deals with the very first steps. We need to have good technology to move forward, and we will need to have more and more better systems to move forward. So now I would ask you, Hayato, to jump in for a presentation, please. And Yvonne, please stay with us.
Great. Can you hear me? Can you see the slides?
Perfectly, yes.
Yes. Great. So I will start. Bom dia, everybody. Obrigado, organizadores, por me convidarem para a conferência. So this is my. So thank you very much. I hope I'll be there in person in one of these coming years. So I'll be talking about vaccines for safer world and pitfalls for development for vaccines for NTDs from a funder's perspective. So and I'll be giving a little bit about the introduction of what we do as a Global Health Innovative Technology Fund, or GHIT Fund, which I manage the entire portfolio and investment process. So we and then I'll be giving a little bit of the one of the investments that we have recently made in terms of these vaccines in the NTD space. So GHIT Fund, we do have a vision of a world free of neglected infectious diseases that affect billions of people. And we are trying to leverage the Japanese innovation in collaboration with innovations outside to solve neglected infectious diseases and free that from the burden from the people who are suffering from them. In fact, what we do is we do make investment in R&D, and so that the product development would have this cycle which would stop the onset of different diseases and increase the productivity of people in those nations who are suffering from these. Just to start off with where we're coming from our funder, we are quite unique in a sense that we're getting funding from both the public and private sector. From the public sector, 50% coming from the Japanese government, and 25% were funded by the Gates Foundation and the Wellcome Trust. So the foundation's providing us 25%, and then the rest 25% coming from private sector. And this particular model of public sector and private sector matching the fund is actually being replicated in places like Korea. There's a Right Foundation, which models exactly the same, and also you're probably familiar with CEPI, Coalition for Epidemics Preparedness and Innovations, which also work with this model, public and private partnerships. So we have invested over the last 10 years over $300 million for product development in the area of malaria, TB, and neglected tropical diseases, which I will talk a little bit more on. But as you can see, our interventions vary from drugs, vaccines, and diagnostics. And of course, vaccines is our main topic of discussion today, which covers about a quarter of our portfolio. And we do cover everything from discovery to clinical end. We have partnered with over close to 200 partners around the world. And you can see how we have some presence in Brazil as well, with work with Bio-Manguinhos in one of the drug projects that we have. I'll skip the slides for now very quickly, but just not only do we do R&D, but we do actually connect with access and delivery, which is going to come to this the thing about this valley of death, which our previous speaker has spoke a little bit about. Of course, NTDs in vaccine space, we do work, we have funded multiple projects, and in the space of Chagas disease, dengue, leishmaniasis, and leprosy. These are the NTD vaccines that we have funded up until now, ranging from preclinical or lead optimization stages up to clinical phase 1 or 2A for the case of leprosy, and we are actually seeing a lot more promising projects coming through. And just referring back to our previous speaker, Yvonne, with respect to the Valley of Death, I think there are multiple valleys of death. I think it was a very good start that we started this session with the translation R&D gap, which was very well elaborated by Yvonne. But I think there's also other types of valleys of death with regards to approval or policy of introduction challenges. Of course, vaccines, they have to be injected into healthy individuals, and it does carry that additional usability burden, etc., for introducing these interventions on the ground, and also the valley or the line that you see at the bottom, the insufficient funding. You know, unless you have the funding for doing these interventions, there's always that you cannot advance some of the R&Ds that you would have. So there's that inherent valley that's at the that underlines all the factors, as well as the third valley being the access and delivery challenges. Where even if you have the product that's already registered, how can you actually bring it to the ground? And that's another valley that I will touch upon a little bit. Just going through the first valley of death, translation R&D gap. Of course, I'm not going to belabor the point that Yvonne has mentioned, with regards to the complexity that's actually there, coming from translating from R&D space to actually moving into product development stages. Of course, it's very important for us to prioritize an early vaccine development pipeline, identify the gaps and align the target product profiles, which we really do emphasize for that investment that we make, to know where you're exactly targeting. And if you don't know where you're targeting, obviously you're not going to be able to make the product where you're aiming for at the end. So not only just start with the R&D, but have the end goal in mind is a key to jumping over this valley of death. And with regards to second valley of death or approval of the policy of introduction challenges, of course, there are multiple regulatory agencies around the world, and each regulatory agencies around the world, not only the FDA or EMA, but also the WHO listed regulatory authorities in how you're actually going to be approving, especially for NTDs, if they're endemic. If they are in endemic countries in different regions, you have to know about their regulatory cultures or regulatory procedures in order for you to navigate that space to successfully enter some of these products going in. With regards to funding, of course, we're only part of the puzzle. We do make funding for NTDs in vaccines, but someone like us just being able to fund product development, but also looking at some innovative financing mechanisms like such as the PRVs or priority review vouchers, or knowing advanced commitments of the vaccine purchases by different entities is also another way of looking at ways to tackle these insufficiencies of funding. And of course, access and delivery challenges is a whole talk in itself, but you have to be able to understand, even after you have the regulatory approvals, how you're going to be moving from that technology that you have that's been regulatory approved, but then bringing into the different countries, because you do have not only if you have to consider the regulatory approvals, but you have to think about the usability and how they're going to be procured, stockpiled, and delivered onto the ground. So there are multiple things to consider ahead of time. Now, let me go back to a little bit of the sciences in terms of what we have invested in this leishmaniasis vaccine. In the world, currently, there are 3 major pipeline of vaccines for leishmaniasis, of which you can see, 3 of which you can see here. The one that I'm going to talk a little bit on is the what you see in the middle, the centrin deficient leishmanin vaccines, the live attenuated vaccine by Genova Biopharmaceutical in Japan in collaboration with Nagasaki University in Japan. Of course, you know very well about leishmaniasis and its impact. You can see the geographic expansion of how these are shown in the world. And this is, if you have a vaccine tool, even with the 50% blocking vaccines, it's modeled that we'll be able to substantially decrease the amount of the VL that's endemic in the world. Even with the 50% blocking, you can actually go down with that level of elimination. And this most effective vaccination strategy so far, at least this is what our grantees claim, is it is possible by having these live attenuated vaccines, live attenuated leishmanin, where they would have the centrin gene taken out using CRISPR technology, where they would be able to achieve this genetically modified attenuated parasites, which is not going to cause infection, but you'll be able to have protection. So it's an old technology brought to life again, but using some of the gene editing technologies that are more cutting edge that weren't available back in the days, so to speak. It is a genetically modified attenuated vaccine. It's on the verge of getting into phase 1, but they do have advantages over the other approaches. And let me show you a little bit about some of the initial animal data, so to speak. So centrin gene, you're just removing centrin gene. For those of you who are participants in this panel, you know about the CRISPR-Cas9 technology. You're basically editing the gene out, and then you're doing the in vitro growth for this wild strain and the centrin deficient gene, and then you're making sure that with the whole genome sequencing, you make sure that the centrin gene is taken out, and you are immunizing these centrin deficient leishmanian parasites onto the animals, and then you're trying out with this sandflies. Basically, you're infecting them using this particular vector. And what you see here, this animal model product, what you see on the top left, the immunized mouse versus a non-immunized challenged mouse. You can see the weight loss being seen for the hamsters for visceral leishmaniasis. And then, of course, you can also see the cutaneous leishmaniasis in mice by looking at some of the lesions that are being affected, with the comparison being shown on the bottom right in the graph there. So just as a summary of this particular study that was done by Professor Saroskar in Ohio State University in collaboration with Nagasaki University and Genova, who is basically doing the manufacturing of this centrin deficient parasite, is that it is safe, and it has been shown the immunogenicity with the high efficacy with, as I said, it's the second generation of leishmanization, bringing up to speed, basically combining old technology, but combining with the new gene editing technology onto NTD space to create this particular parasite. Now it's planning to be tested in phase 1 in the coming months. So just concluding, I kind of rushed through this, but just coming back to this, of course, we talked about the valley of death, the translational science valley of death, as well as there's a second valley of death about the regulatory valley of death, which requires alignment of the regulatory policy financing continuum. But there is that portion of the financing valley of death where that sustainable vaccine development will be key by using some sort of a collaboration. And of course, we, as ourselves, GHIT Fund, we're just one piece of a puzzle, which we would welcome any of the participant who is in this room to contact me if there's any interest in our work, as well as any potential synergies that we obviously observe for some of the programs that we see. Because we do make investments in not only vaccines, but also in diagnostics and therapeutics, we have seen some programs being able to be bunched up together to tackle the program together. For instance, the leishmaniasis vaccine that I mentioned at the end, that particular technology is being applied for leishman skin test as a way of testing whether the patient has been subjected to has had leishmaniasis before, being able to use that as a diagnostic as well. So we're trying to look for innovative ways to combine our technologies together. This last slide here, just, I don't know, maybe it's just a self-promotion in a sense. If you're interested in finding out a little bit more about us, please go to this QR code. If you sign up on the newsletter, we'll be able to send out the next request for proposal, which is going to come out in June this year. So with that, I'll acknowledge Professor Ape Saroskar for his work on some slides, as well as my colleague Cecilia, who has helped me create these slides. So with that, I'll pass over back to the chair, and muito obrigado.
Muito obrigado, Hayato. Vejo que o seu português está bem avançado.
Has improved remarkably, and thanks for your presentation. I'm going to give the floor now to Paulo Negreiros. You can make your presentation from the pulpit, from the lectern over there, and we'll move on to Q&A right afterwards. Good morning, everyone. It's a great pleasure to be here with you. I'd like to thank you at the Getúlio Vargas Foundation and Fiocruz for the chance to participate in this symposium. We have a research problem in technological learning and industrial industrial knowledge. I've been a leader at the Getúlio Vargas Foundation involving the management of the process of innovation. It's a set of startups in Brazil in the area of biotechnology for human health. I've had the privilege to lead that study with a group of researchers that are highly talented, involving bringing together, for example, thank you, bringing together Professor Daniel Oziel from the Federal University of Rio de Janeiro, Marcela Cohen from the Federal University of Fluminense, Pedro Gomez, a master's student at the State University of Campinas, and the assistant researcher at the Getúlio Vargas Foundation, Lohana Palini, postdoc at the Oswald Cruz Foundation, and Bernardo Cabral, a professor of economics and biotechnology at the Federal University of Bahia. That is, we have an extremely varied and distinguished group with contributions that are very important to this study. The motivations for this study mostly involved facts that even in the advanced economies, investments at risk, high risk investments have not been geared to the science and health studies. They've been geared to digital technologies and thereby avoiding risks in the health arena. Even accelerators have, in a way, they haven't felt so attracted or drawn to the area of health sciences. Hence, many ventures and startup initiatives have not gone too far due to lack of investments. And certainly also in the literature on innovation, there is a scarcity of studies involving the process of innovation and biotech startups and how they may evolve till the stage of commercialization, successful commercialization. This scenario has been aggravated, has been worsened in the context of developing economies, which is the case of Brazil, in which we have over 200 million, and we have a demand, an increasing demand for public health services. And we have, because of the universalization of the public health system in Brazil, there is an increasing demand for therapies and biotechnological therapies and medications, and certainly for pharmaceutical inputs. At the same time, the country has a very low supply of pharmaceutical inputs and biotechnological products, which leads to high imports, large volumes of imports. And that has been even greater at the time of COVID, a greater dependence of imports, leading to a commercial trade deficit of over 7 billion reais in Brazil. And even though we're the sixth largest pharmaceutical market, we only produce 5% of its inputs. We produced 80% of them in the past, but we have fallen down in that production. There is an urgent need to better understand the ecosystem of biotechnology for human health, to try to see how we can strengthen it. That's why the objective of this study is to understand this process of accumulation of capabilities to produce and innovate in biotechnology for human health in the Brazilian ecosystem for health and biotechnology, especially at the level of startups. The analytical model of the study basically involves three large variants. The central variable basically is the level of capacity, technological capability, which is a set of resources that will allow companies, startup companies, and organizations and institutions to undertake production, and also innovation. That's a central variable, and we would like to understand the sources for these capabilities and the results, the outcomes, the impacts that the accumulation or lack thereof of these capabilities have generated. Basically, our study has involved a set of 18 startup companies in four large areas: in vitro diagnostics, startup molecules, gene therapies, and tissue therapies. And they have a time they've been in operation since 2008. Some of them are younger, 2021, for example. The names have been codified because the startup companies, their names would not be publicized. We have an agreement with them. We have a very interesting set of startups, even though the sample is not too large, but the study has been in-depth and they have offered rich evidence, wealthy evidence to illuminate public policies, to shed light on public policies. They are scattered around the southeast and south region of Brazil, especially in the state of São Paulo. And the study started in 2021 and involved a series of steps going through recursive processes, coming and going, and so on and so forth.
So that we could obtain evidence, in-depth evidence, and qualitative depth also is this. And the academic studies that review literature they obtain from the companies, the information, and the final, they generate a paper that will be delivered to society. So we seek to involve these startups since the beginning of the study so that they could help us with the design and the questions of the research. So we made several workshops with them to feel the problems and the main issues and their pains, and that this could be embodied in the design of the study. So they followed through all the study, which gave us better connection with this important side of the demand. In terms of business model, these startups are distributed among several different business model, also the hybrid ones, which involve product platform and products, and their investments are basically that come from governmental bodies such as the Development Bank. And if we opened that part of the investments, we will find those that come from the state agencies for innovations, such as Embrapi and FINEP. And we also have multi-strapping investments. So going to the main variable, which is regarding technological capability, we understand this is a storage of knowledge that the institutions accumulate to generate innovative action. So there's a set of knowledge that distributes, such as human resources skills, business skills, professionals, and technical and physical systems, and so on. In the case of the startups, we prevails the human resource in the researchers and the professionals, and the others are more well distributed in the ecosystems. And the idea when we examine the technological capability is not if they have or not, but the degree in which it exists. So this is why we measure the level of technological capability in these startups. So we avoid the conventional proxies in literature, and we identify this maturity model that is based mainly in the revealed capabilities. That is the capability of the startups revealing their activities and what they're able to do. Another distinction is where you differentiate the production and innovation. One thing is producing of vaccines with an existing technology and you're a user of this technology, and the other thing is to develop the capability to adapt this vaccine and even more scale up and create a new vaccine. So we identify these levels and up to which point these startups develop themselves in the capabilities. These are the results, and from these startups 2 and 4 reach the levels 2 and 3, so they are able to adapt in existing technologies, and with a new degree that is for themselves in the regional context, but 7 of them advanced to the advanced level next to the international frontier. But here we have, gentlemen and ladies, 5 startups that could reach world-class level. and 2 of them went to the commercialization phase. It's a great merit considering the concept that we have, the hardships that they face to operate in a country like Brazil to reach this level, carrying out activities that are new to the world in a very difficult area. So we have much to learn with these startups. They generate interesting evidence to public policy generation and how we can support these technological capabilities and not allow to these levels weaken themselves and get lost in the ecosystem. So we should also look at these fives that could reach this level 5, and some of them are hybrids, and the type of investments that they receive are mainly for FINEP, CNPq, and FAPs. And in terms of the partnerships that they establish, and they carry out many partnerships that are local, universities that are local in Brazil, also with specialized consultants and private international research centers. So the localization of these partners are Latin America, followed by North America, and this axis is from where they seek the knowledge to they can carry out these capabilities. They use external sources for such as training abroad, partnerships. Some of them are based in research and development, some are not, but they also carry out internal efforts for knowledge creation. So going to the results and the outcomes and the levels of these 5 startups, we have outcomes in terms of patenting activities, some in the US and also in Brazil, and also results in terms of technological activities in the production and markets. So in the end of the presentation, we're going to compare those that were able to achieve this commercialization stage against those that were not able to do so. So in terms of partnerships, they all can make partnerships with specialized consultants and also with local universities, but there's a difference. Those who could get to the commercialization phase, they have very important partnerships with pharmaceuticals and biotech institutions, in addition to having these partnerships with the local universities and specialized partner consultants. So there are clues that you can observe to get to the commercialization phase. So the regions are Latin America and North America, and the efforts that they make are basically of international PhD, international collaboration, but also local and also internal efforts of knowledge generation. So we can see a distinction in terms of efforts of learning that for those that could reach their commercialization against those that could not reach this level. So regarding results obtained in this commercialization stage for respectable patents in the US and Brazil, and they could also reach market outcomes for these that have reached this level. So these studies allowed us to issue some recommendations in terms of ensuring the needed funding for them to generate the innovative activities, the investment in practicals, in learning practice to get to this level of commercialization, get to higher levels of technological capabilities. Also, these develop partnerships with biotechs and pharmaceuticals is also very relevant in the process, and the development of capabilities beyond technical and scientific. So technical and scientific is essential, but it's not enough. So there you need to develop the skill, management skills, marketing, entrepreneurship skills. So adjusting the business in itself, that's why it's important, this partnership with the management training. For public policies, we strongly recommend the simplification of access to funding, which was an issue that was very much discussed by the startups, the difficulty of finding pathways, of the lack of integration among the organization that operate the fundings, and also consider the capacity of the innovation of a main activity in this process, also to support startups to go through the go up to the commercialization phase, and also to expand this commercial skills. So I'm here. Thank you very much. Thank you. I'm available to answer your questions. Thank you very much.
Muito obrigado. Acho que é excelente, né?
Paulo, thank you very much. The study that you carried out is excellent, and the previous organization who seek these spectrum of different initiatives, and we're having a tight time here. We should end at noon, but I will ask a little bit of patience. So we have 3 minutes to have the opportunity to have 1 or 2 questions to the panel. We have one here, and the questions that we received online, we are transmitting directly to the speakers, and some of them are very specific, that I think that they're of much more of specific interest of one of the other. So thank you very much. Congratulations for the 3 speakers. And Paulo, I have 3 questions for you. One is the role of the meetings in these studies, the nucleus of technological information, and we showed that 56% of the startups were in São Paulo. And when you separate these 5 that reach the global level, how is this divided in São Paulo and the other states? And the other thing is the degree of investment that was done. And if you have this number, and I know that FAPESP's role is a key role compared to other states, and the level of participation of the students, especially of postdoc in these startups. This is a great question. The NITs NITs are they belong to the federal university, the public university, especially in the southeast region and in the south region of Brazil, and they participate in these partnerships with the startups. And regarding the specific values, we do not have this evidence, but it is an important evidence to be sought in future studies. And regarding the participation of students, yes, in some cases we were able to find a participation of students, but still very much reduced. So this would be a suggestion for the public policies, the involvement and the fostering of the researchers in this sense. Regarding these 5 startups, the great majority are in the state of São Paulo for sure. So, and they reach the highest levels of the study. So unfortunately, we have to close our panel.
Colleagues, for being with us online. And we will direct to you any further questions. I received some specific questions that I instructed the team to direct to you all. And I'm pretty sure that we will have some follow-ups from your presentations for all the 3 of you directly from my team here, at least. Thank you all for being with us. Muito obrigado aí a todos, e a gente ainda tem mais uma sessão aqui. Obrigado.
We still have one session. Thank you very much. Thank you very much, Ricardo, for your moderation. Thank you, speakers, and thank you all. Well, moving on forward to their program, we invite as chair of our Promega panel, the Vice Director of Bio-Manguinhos, Marco Alberto Medeiros. He's a doctor in biotechnology by the Federal University of Pelotas. He has a postdoctorate in the University of California in Los Angeles and Pasteur Institute, and he acted as invited researcher in the vaccine developer in GSK lab in Vienna, Austria, and we also invite Promega Corporation senior researcher Juliano Alves, who will make the presentation platform of development of rapid and sensitive methods for analyte detection. He's a doctor in pharmacology by the Federal University of São Paulo. He was a researcher of the Genomic Institute for Novartis and also Script Research Institute. At Promega, he worked with the development of several technologies used in the breakthrough of medicines. The group leader in the Institute of l'École Normale Supérieure, Lionel Navarro, who will give his presentation, Plant and Microalgae Extracellular Vesicles as Carriers of RNA-based therapeutics. So we remind you all that this presentation has confidential classified data, so it will be interpreted for those who are online. For those who are present, we ask you that do not use your smartphones either in video or photos or any kind of recording. So he has experience in the study of interactions, pathogens, and hosts, and innate immunity, epigenetics, and silencing of RNA. His research team was pioneer in the role and the regulation of small intracellular RNAs in the interactions with gas hosts and bacteria, and the identification and characterizations of bacteria suppressors, and also in the control, transcriptional control of immunological for this methylation of DNA. So he founded several companies and spin-offs developing disruptive technologies to control infectious diseases of plants, animals, and humans. So he's currently director of research for Natural Recherche Scientifique in France. Scientifique. So I pass the floor to Marcos Alberto Medeiros.
Excuse me. I'd like to thank you, Lionel and Julian, for accepting to talk in this event, the International Symposium. Yeah. I hope you enjoy your lecture. Go ahead, please.
Boa tarde a todos. Primeiramente, eu gostaria de agradecer.
You all, I would like to thank the opportunity of being able to talk to you all. My name is Juliano Alves. I am a senior researcher at Promega Corporation. I'm going to talk about a set of solutions driven towards the immune messengers, and we talk about assays. We think about the ELISA assay, and we think, you know, that the time that is required to generate after the cleaning of the wells and the regulators, and Obviously, the time consumed with the proceedings are taken since you take several hours to have the results. So thinking in a way of solving this process and to simplifying it, we thought about the Lumit platform. So can I change this? I'm not going to take the risk. So in this Lumit concept, these assays, to simplify, they consist in two primary antibodies that are combined with another substance, and they, in the presence of this analytes, they unite and they conduce to the complementation, the activation of these analytes. So in the substrate, you have the bioluminescent results. One of the advantage
ao invés de você ter todas aquelas múltiplas etapas, você reduz o número de passos necessários a gerar o seu resultado.
You reduce, consequently, you reduce the time of incubation, and ELISA can lead several hours to obtain results. And this Lumit assay, the time is reduced from to 120 minutes, 2 hours. One of the advantages is the high sensitivity for those researchers that work with cells. There's a possibility of reducing the number of cells per study, and the assay window it's better, and also the development.
Do ensaio, você pode correr testes.
The possibility of we usually say that it's a homogeneous assay because it doesn't require the cleaning of the blades. You add reagents, you do the reading of the results, and it's easily adaptable to the heart. Once I've said that, we can envision a series of applications, possible applications for the Lumit platform, the detection, direct and indirect detection of analytes, protein-protein interaction studies, development of competition assays, and also the detection of antibodies. When it comes to the direct detection of analytes, the main requirement is that there should be two primary antibodies, and these antibodies should not be of different species. The advantage, as you maximize the number of antibodies that may be combined in order to select the pair that will be ideal for your assay in a short span of time. Using this approach, today we add many solutions, as you see on the screen, interferonogram and 2 and 4F alpha, as well as hereditary factors like VGF or insulin. But also there is a possibility of researchers generating their own in-house method through the use of assay kit, which has all the necessary components to conjugate your antibodies of interest, including fragments of the luminiferous. If the researcher does not want to generate his or her own reagents, the other option is to do the pairing of the antibodies of interest with secondary luminous antibodies. These antibodies, I can tell you, they come in different flavors, like anti-mouse, anti-rabbit, anti-goat, and anti-rat. The difference in that's adaptation is that you didn't have to do the conjugation, as I said before, and this is an approach that's used currently in intracellular events like phosphorylation and cellular degradation. Another possibility of this platform, as I said before, is that it allows for a study between protein-to-protein interactions with the use of antibodies that link to purification tags, such as this tag, plaque tag, or GST. This tool is used, for example, in screening of drugs like Protex that induce the proteolytic destruction of unregulated proteins in cancer cases. Another possibility also is the development of competition assays in which a tracer molecule, be it a organic compound linked to a luciferase fragment or an antibody, for example, it's dislocated through the interest analyto losing luminescence, for example, bioluminescence. An example of the solution developed by Promega was this SCIN, in which you detect antibodies that link to the arsenal, receptor. Now, this methodology can also be used in assays for the identification of metabolites. The last example I'd like to bring to your attention is the possibility of using this platform to detect platforms, be it for the confirmation of the exposure of individuals to certain pathogens, or to try to visualize the potential of neutralization of these antibodies in human samples. And we can use these tools also in the discovery of antibodies with therapeutics and antiviral activities. To exemplify what I just said, in the recent past, we've developed a LUMIT biochemical assay that consists of the interaction. My pointer is not working too well, but I'll try to explain it to you better, step by step. We have developed an assay some time ago. It consists of the interaction between the RBD portion of the spike protein on SARS-CoV-2 with the soluble receptor. How does the method work? In the absence of an interference or an antibody or an organic molecule, these two proteins find themselves in a solution. They do protein-to-protein interaction, and these protections have an FC tail derived from an antibody, and that allows for the use of secondary movements of antibodies that will lead to the reconstruction of analog luciferase and the generation of bioluminescence. The absence of an inhibitor agent, you have 100% luminescence. And when you have an antibody or a molecule or something that interferes in that interaction, the RBD will be sequestered and it will be kept from interacting with acid 2, and surely, consequently, limit antibodies won't be able to get close to the luminescence. The loss of bioluminescence in this case is a result of the presence of an antibody or a molecule that affects in that interaction. To demonstrate the concept and to prove the concept that this methodology works, we've done a limited screening to antibodies commercially available, and we can ascertain the fact that in the first part of the graph, and the samples where we didn't have any bars, these are antibodies against spike protein and SARS-CoV-2, and our method was able to detect these antibodies with a neutralizing potential, and the control antibodies didn't have any interference and the interaction between RBS2. It's worth highlighting that the bars in the middle of the first graph are cut in half. These are antibodies that were generated in the first outbreak of SARS-CoV in 2003, and they're able to interfere with up until a certain point. We have tested also sequences generated with in silico predictions based on the interface, interface region between the acid2, S2, and RBD receptors, and we tested to see if we would be able to detect this interference. We confirmed the remarks published at that time that 3 sequences that the authors called mini proteins were able to interfere with the RBD S2 interaction. They demonstrate the potential of this platform to be used in discoveries of antibodies with therapeutic potential, in this case, antiviral activity. But it's not only that. You can do many studies, molecular studies. In this case, we looked into the role of the mutations present in RBD and the process of interaction with S2. Various mutations were reported on and many variants. And the concept is, depending on the affinity, probably you will need more analyto to be able to interfere in the RBDS2 interaction. To mutants that we have tested, we noticed that 3 mutations caused a dislocation of the curve, a displacement of the curve, which suggests that these mutations conferred a certain increase, a certain degree in increase in affinity as for S2 vis-a-vis S2. In addition, we looked into whether these mutations would have a role in the process of evasion of the immunological system, of the immune system. That is, if they would prevent the linkage of the antibody. And we had tested a small panel of antibodies. We went back to that panel. We generated RBDs with FC portions, so we could just replace them with the RBD that we have in the assay. And we noticed that for 2 antibodies that led to complete neutralization, 2 mutations were caused. They caused the complete loss of ligation. And it's worth noting that one of these mutations, N501Y, has a double role. It plays a double role. It apparently increases affinity with S2, but it also inhibits the linking of antibody that was previously able to neutralize the interaction between RBD and S2. And we looked into mutations of variants coming from sequences coming from variants of SARS-CoV-2 from the United Kingdom, South Africa, and Brazil. We noticed the same effect, since these 3 variants have that mutation, which reinforces the remark that this mutation can lead to an escape of the immune system. When it comes to human samples, we did an analysis of samples that were positive, confirmed for PCR, and negative samples, so as to observe the distribution of those samples in order to ascertain a cutoff that could help us when we would test unknown samples. When it came a time to test unknown samples, as we can see in both cases, third samples of serum and plasma, and we could notice that negative samples began to group together below 30% at a neutralization value of 30%. As for positive samples in green, we noticed that these samples, most of them tend to group together, to come together between 80% and 100%. With this information, we have defined a cutoff line and 30% neutralization so as to distinguish between positive and negative samples, and that led to a performance of a sensitivity of roughly 80% and a specificity of approximately 100% in these cases. With this criterion, the way we have defined it, we ran a temporal analysis of samples of patients that were infected by SARS-CoV, and we've tested several samples throughout time, and we noticed that the antibodies that were able to interfere in the RBD and S2, they began to surface and on the 12th day, and these individuals that were affected, the antibodies lasted up to 6 months. So similarly, we tested also our methodology on patients that were immunized with the coronavirus vaccine, and we noticed that only those individuals that were immunized after the 2nd dose, there was a spike in the immunization. The sample was able to completely eliminate the bioluminescence signal in our methodology. These results were published recently, and recently we used this information, the lessons learned in the course of the pandemic, to expand and try and develop new serological solutions using the GHIT platform, and 2 neglected diseases that we are keen on developing are Zika, because of the historical background, and dengue fever, and because that is a problem, a public health problem in Brazil and so many countries, and a concept of the assay, the design of the assay is a bit different because in this case we used antigens related to nospherase fragments, and if there are antibodies present that recognize that antigen, that antibody during the linking process will complement nospherase, generating a bioluminescent signal. And preliminary results we can observe in the graph on the right side of the screen, on the left side of the screen, excuse me, several antibodies allowed that we were able to detect several of them, which makes us see that we can use that platform for other purposes. Different volumes of reactions can be used, showing the flexibility of this platform and miniaturizing and maximizing the use of the reagent. But we also know that serological assays for the detection of antibodies against arboviruses, there's a problem of cross contamination. We use polyclonal antibodies for dengue. Our kit was able to detect, that's the first graph, to mitigate this problem. We developed a blocking agent that was able to neutralize the presence of this antibody, contaminant antibody, but most importantly, the presence of this blocking reagent didn't interfere in the reaction and the identification of Zika antibodies. It's still in progress. And parallel to it, using the data from the same concept, we're developing a test to detect anti-dengue antibodies following the same principle, using antibodies antigens. And preliminary results we were able to detect. And in this case, the same problem surfaced when it comes to Zika, but we were able to develop a blocking agent in this case that inhibits a crossed reaction, but it doesn't interfere in the results. But it's still a project underway. It's currently underway, and we'll have more details to talk about it later on this topic. Just to wrap up my remarks, today I presented to you the LUMIT platform, the detection of the interaction between virus proteins and infected animals and their proteins. It's easy to then host proteins. This methodology can be used in addition to a virus neutralization platform, be it immunized animals or not. This molecular studies can be done through this platform to understand the role of mutations in the interaction between these two proteins, and also they can expand it to other viruses of interest. And most importantly, this assay can be automated and miniaturized. To wrap up, I'd like to acknowledge all the scientists involved in the study. We can't do anything alone. We depend always rely on a network of collaborators. And I would like to extend a warm thank you to all the participants of this panel. And now I'm available for Q&A. After Leonel's presentation, we'll run a series of questions after Leonel's presentation. Byron, please.
Pointer.